Okay, so now moving on to our last speaker in the Road to Personalized Medicine session, we are honored to have Dr. Martin Turner, Professor of Clinical Neurology and Neuroscience at Oxford University in the Nuffield Department of Clinical Neurosciences. He's also the co-director of the Oxford Motor Neuron Disease Care and Research Center. And we are honored to have him present Project Ambrosia today and his presentation on the biomarker challenge in ALS, about a 5-year research project searching for biomarkers in the field and leaving a legacy resource to improve diagnosis. Thank you for being here, Dr. Turner, and the floor is yours.
Great. Can you hear me okay?
Yes, we can. Thank you.
Yeah, perfect. Well, thanks for the invite. It's a pleasure to be here, David, and others. So I'm going to talk a little bit about biomarkers and why we think this is really so important amongst all the other great science that you can hear about. So I'm going to show this slide to start with, and it's quite a busy slide, but it's really just to show you that this is a report from the 1890s. And, you know, we can see already without any difficulty that this is a case of ALS. And that's really important to remember, that this is a clinical syndrome that is instantly recognizable. So in this particular case, there's a family history, and we now think a lot more about that. I'll come back to that issue. We think patients are generally quite well beforehand, and that's another interesting point. So we don't find any previous illness. There's this progressive story, in this case, dragging the leg starts to waste, there's weakness, and then in the hand, and then these exaggerated knee jerks, sensation normal. And we also have this fasciculation that you can see on the side there in another case. And that is unmistakable, and that's, despite the fact that it's many, many years ago, there's no doubt whatsoever that that's a case of ALS. Okay, so what Charcot described is the pathology of these lower motor neurons, shown in green in the middle here, branching out into the limbs and the speech areas, in combination with damage to the central motor pathway, the upper motor neuron. And that gives us our characteristic clinical syndrome. But we also recognize that this syndrome overlaps with a dementia, and that's something that is now very well understood. Very few of our patients get a frank dementia, but quite a lot of them will have some involvement of their cortex. The other issue is that within this syndrome, individuals really tend to set their own pace. So the condition itself interacts with a person's body, their architecture of their nervous system, the cellular repair capacity they have, all sorts of factors that we're starting to uncover. And for an individual, the rate of change seems to be quite fixed. And so for someone like Lou Gehrig, very typical sort of pattern of disease, unfortunately progressing within 3 years, right through to the really unusual, but definitely still within the spectrum of people like Stephen Hawking. And of course, extremely unusual, that length of time, but certainly not outside the realms of possibility with this condition. It's just that for most patients, unfortunately, it's going to be a matter of a few years. What you can see on the side is the decline in the disability. That's the top graph. I'll just use the cursor here. And you can see that patients progressing downwards at different trajectories. But if you look at their rate of change on the lower graph, then it's rather fixed for most individuals. So if you say, or my patients say to me, what's the cause of ALS? I kind of say, well, actually, there's not just one. You've got a very long motor neuron here. We can overstimulate it. That's possibly partly aggravating. That may be due to upstream problems. We can attack it with the immune system, this microglial cell. We can upset transport in this very long axon. There's lots of change going on in proteins coming in from the nucleus into the cytoplasm, perhaps not getting back inside in time, damaging the mitochondrion. All sorts of ways we can upset this very, very finely poised balance. And we end up with this sort of model, really, whereby we've got a common pathway here on the right-hand side at the bottom. And that final common pathway there is fed really by this pathology called TDP43, and that underlies most of the cases. And there's a whole range of genes up here that you'll recognize, C9 and FUS and TARD-BP, but actually the vast majority of cases coming from complex pathways, but they all converge on this protein which goes wrong. And then you've also got other rarer forms like the SOD1 mediated ALS, which comes in further downstream and doesn't seem to involve this. But the final common picture is very, very recognizable and would be like the descriptions that you saw in the first slide. So to set the scene, really, we end up with this complex mix in ALS of lower motor neuron involvement, which may be very pronounced, and we sometimes call that progressive muscular atrophy, a very slow process. It may be pure upper motor neuron, in which case we call it primary lateral sclerosis, and it may be very pure frontotemporal dementia, and that's also a slow process. So at these extremes, you have really quite slow conditions, but when they get mixed in the middle, which is more common for most patients, there will be a much more rapid progression. And if it mixes with frontotemporal dementia, it's among the most aggressive, although that's not the commonest. So the reason we need biomarkers is because it takes an ALS patient, or MND as I've written here, it takes 10 minutes usually for me to make a diagnosis, and often I can make it from the letter from the primary care physician, but that person would have taken a year to get to me. And that's for complex reasons, but it's a very, very big problem. The other issue is that in a clinical trial, here's the disability score on the side here. These are all patients in a clinical trial, and their trajectories are all very different. These people here progressing very fast, these ones much more slowly. Now, to make sense of that, to decide if a drug is working, is extremely difficult. And this is the problem. It seems to me unacceptable that we have any more failed drug trials. We have to try things, but we've been trying things in large trials without any biomarkers, nothing to measure, and we've been doing that for 30 years or more. And out of that has come riluzole and edaravone, both with very small effects among a huge number of drugs that have been tried. And yes, one of the problems is the drugs may not work the way we want, but one of the big problems is we've got no way of measuring their effect without doing very long studies that take 12 to 18 months. So what could we get from something we could measure, we call a biomarker? Well, we could have patients earlier into studies. So before they're disabled, at a later stage of disability, we can get them in earlier, and we can also get some of those more unusual lower motor neuron cases in. We can also stratify patients much more. We can decide really and model the data on people that we already knew really were on certain trajectories, and that allows us to be much more sensible about how we analyze the data and not expect a drug to show an effect on every patient. And a big prize really would be something we could measure that would be more sensitive than simply saying, are you less disabled than we'd expect you to have been at 12 months, which is a very blunt instrument. So something we could measure. And that might be something we could measure that we could say, look, this drug's not working, let's try something else. Now, the reason I put this poster on the side here, this is from the 1980s in San Francisco, and it was telling people to take their AZT, which was the only drug available. It wasn't highly effective for HIV, but many of those patients who did take that drug, they did stick around for the cure. And I think, you know, we've started at a low level with riluzole and edaravone, but the idea is that we're making rapid progress, and with these biomarkers, we can move that forward. So we have a signature. And in the cells, we have this protein TDP43. It's a normal thing. It should be there. It should stay in the nucleus, but it comes out. That's part of its normal function, but it shouldn't come out and then get aggregated like this. Now, it's complex why that happens. There's a huge number of reasons why, but that is the core signature of this condition. That is the diagnostic test, but we can't measure it. It's very, very challenging to find. It's not really in the blood to any great extent, and it's not really in the spinal fluid, and it's too traumatic to take a biopsy from a patient. So this is very much the focus of the very, very important cellular science, but it's not our readout. So it struck me that really the approach that I thought might work was to take patients and to follow them over time, because that was really the thing that hadn't been done. And I thought, well, what tools can we use really to look at the whole body, the whole system, the whole nervous system? And MRI, as I'll show you, is a way of looking at structure and function. We can look at spinal fluid, which you can see a sample of there. And, you know, patients will be able to sort of tolerate having an MRI scan, but they just need a little bit more help than is standard in clinical practice, but it's doable. So I undertook to do these studies in patients serially every 6 months. Now, this is the original patent for MRI. That's from the 1970s, and it's come a long way since then. The original pictures of patients with ALS, you can see here, really were a bit distorted, a bit grey. But you can see actually there were some changes in the corticospinal tracts, and that's from 1988, so 30 years ago. But fortunately, we can be much more sophisticated now with imaging. And we don't use the MRI scanner so much to say, well, does it look like a brain? Because the brain looks pretty normal in ALS when you just look at it from the outside. It's not particularly shrunken. It's not like Alzheimer's. But we can actually look in much more depth inside the tissues and look at the individual tracts in the brain, and also how the brain talks to various parts of itself, what bits are talking to each other simultaneously, and so build up a picture of the brain to do with structure and function. Now, when you do that in ALS, we get a signature at the brain level. So we've had a signature at the cell level, and the signature at the brain level is these white matter tracts, the descending corticospinal tracts, and particularly this bridge tract between the two halves of the brain called the corpus callosum. And the reason I've lined this up like this is because this is a study that I'm showing next to it from 1960, and this is post-mortem brain. And what Marion Smith was doing there was looking at slices under the microscope and seeing where the changes were. And you can see that we're seeing these changes in living people now. So we're doing in vivo living pathology studies, and that's exactly what we needed. You know, we need to get away from the end stage pathology and much more into what's going on in the living person. It does seem that certain parts of those motor tracts seem to be rather vulnerable and maybe a marker really of the sort of aggressiveness of the disease. So that's another area that we're interested in, this particular part of the motor tract called the internal capsule. And actually what we found over time was that when someone comes to diagnosis, that all these white matter tracts are pretty damaged. And that's, you know, really shocking. But it's partly because they've had a delay in diagnosis, but it's partly because the symptoms really don't appear until a lot of damage is done. That's a very difficult aspect of degenerative diseases, but it seems pretty clear that that's the case. Over time, we see more changes in the grey matter, and so that seems to us to be the marker that you would use more in a trial. This is showing some data where we just asked the brain scanner to tell us what the sort of brain activity was at any one point using the MRI scanner, with a person lying in the scanner doing nothing, and could it separate patients from controls just on the basis of the fluctuations in the oxygen levels across their brain, and that's called resting state functional MRI. And you can see that with several regions of the brain, it's possible to demarcate patients very nicely from the healthy controls. And this is the sort of way that we're starting to think about the brain going forward is in terms of function. I want to touch on fluid markers of damage. So another way that we can approach this is to look at the breakdown products of nerves. And what you can see here is that when nerves are damaged, they release these neurofilaments into the spinal fluid. Now, if you measure those in patients, in the spinal fluid, you see that the level is much higher in the ALS group than the healthy control. And in fact, actually, when you take diseases that are not healthy, but they're not ALS, you also find a pretty good separation. So this is looking like actually quite a helpful marker for us. Although it's not a specific thing to ALS, you find these neurofilaments in all sorts of conditions like Alzheimer's and other conditions. It's not quite so good in the blood. You can see that the difference between patients and controls isn't quite so good. But remember that we wouldn't simply be doing a blood test in a blind way. We would have some information from the patient. They would be telling us certain symptoms they've been having. And so a blood test really as part of the diagnostic process is something which is within our reach now. One of the key things was my patients who volunteered to give me their CSF, their blood, spinal fluid over several time points, allowed us to make this discovery really that the level of the neurofilaments in the spinal fluid strongly reflects the rate of progression of disease. And that's a very valuable marker for a trial, because it means if we give a drug and we perhaps lower the level to come down to this level, it means that we get an early readout that the drug might be working. And that's helpful then for an individual, the level stays the same. So we're not sort of worried that perhaps it was going to go down anyway in that individual. So this result, which only came from massive efforts of patients really having serial samples, has really driven neurofilaments forward as a major biomarker for trials. Now, this is a bit hot off the press, really. I can't give you a lot of information yet because we haven't done all the analysis, but I've got a junior doctor who's doing a really interesting study at the moment and actually saying, well, look, let's get this into the real world. If I take a blood test in the clinic, is it helping me diagnose the more difficult cases? And there's some early positive signs that that is the case. And even more interesting, but a little bit separate from ALS, is actually this might be quite an interesting test for a primary care physician looking to see if someone's got a neurological problem in a more general sense. It's certainly not going to be something a primary care physician could use to diagnose ALS, but it might actually turn out to have some value in at least getting people to a neurologist faster because there's something going on. So that's an exciting study that I hope will have some results in the very near future. So what about the deeper content of the spinal fluid? Well, we can separate spinal fluid, like many fluids, into a huge number of proteins using something called mass spectrometry. This is extremely complex biochemistry, and I'm lucky to have colleagues who understand it and can help me use it. Now, when we do that in ALS, we find that these markers here came out completely unexpectedly in the spinal fluid. And these are things called chitinases. They're secreted by macrophage immune cells. And again, they seem to be very raised in ALS and seem to separate from other diseases and different conditions like Parkinson's here, disease mimics, healthy people, and also PLS patients are a bit lower as well. So this is a new type of marker. It may be a marker of inflammation in the brain. That may be something which we want to encourage because it slows the disease down, or there may be aspects of inflammation we want to slow down because they're aggravating the disease. We know that inflammation happens in the brain in ALS. This is some work I did now more than 10 years ago, looking at microglial activation in the brain using a PET scan. And you can see that it's activated throughout the brain, including in this patient here who had predominantly right-sided symptoms, and this is the left side of their brain, which is what you would expect, the motor region opposite the side of the body that's affected. So we've now got a biochemical marker of that, and that's something that we're working hard on to take forward. So I want to finish off really just talking a little bit about the pre-symptomatic stage. So, you know, when does ALS begin? Unfortunately, I don't think people are normal before they get symptoms. I think they often notice a sudden drop, and Lou Gehrig did. This is his batting average here. It suddenly dropped off. But the reality is that there's a huge reserve in the body. We know that. And so by the time symptoms arise, there's been a lot of damage beforehand. And the body's been compensating. And there's this period probably of many years for some genetic patients from birth where they've been compensating for damage. And a very famous Canadian who spent some time in Oxford, William Osler, the father of clinical medicine, he made this very important point, really, that we should think about the sort of people who develop a disease, not just the disease. Now, I don't know what the single cause of ALS is. I think it's multifactorial, but I am struck by how physically fit many of my patients are. Not all of them, but generally far more than aren't. And I don't think that exercise causes ALS. I don't think it's trauma in a simple way, but I think it may be reflected in people's motor system and their architecture of their nervous system, may be reflected in their ability to perform sport and operate at an athletic sort of level. But it's a very, very important clue, really, because it gives us some sense of what the mechanisms are. And we can use hospital record linkage, which I've done with colleagues, to look at various things that are happening to patients before they get symptoms. And we find that in general, cardiovascular disease is a little bit reduced as physical activity is increased. We find there is some link to tissue injury, but not a single head injury, not a single event, possibly much more complex. And perhaps it simply serves as an extra stress on the system. It's not the sole cause, but it simply adds to the problem. Occupations, military, professional football, and agricultural work have all come out. But actually, probably in my mind, these are surrogate markers really for fitter people rather than looking for a particular toxin in agriculture or a particular problem in the military or a particular issue in football. And there's some great clues coming now from metabolism. We find that there are lipid changes in ALS patients many decades before the onset of symptoms. They're not lipids that are what we call bad lipids. In fact, they're very healthy ones. They have a good HDL-LDL ratio. But it's telling us that there is a huge metabolic, wider physical, multi-system process going on in this condition. And we've got to broaden our horizons really to think about it. It's not a single cause. So I'm interested really in trying to get back into this area where all this sort of stuff is going on under the surface, the iceberg, until it emerges as symptoms, and then we see this terrible trajectory here for most patients. What's going on in this area? Well, gradually the motor neurons are falling and the cells are decompensating. And what would be amazing is if we, rather than put riluzole and other drugs like this in here, if we could get them in here. Now, how can we find people before they get symptoms? Well, at the moment, we have to go to those carrying rare genetic forms, and they're a small group relatively, but they're about 10% of all patients, and most of them are carrying this C9 mutation, which I'm sure you'll have heard about at some point. We also find some patients who don't have a family history of carrying this, and overall, it's probably about 10% of all cases. Now, if we take people who are carrying these mutations, they have a high chance of getting ALS. It's not a 100% chance. That's very important. It may be actually quite a long way from a 100% chance, but it's definitely causing them to be under strain and their cells to respond. And so my view is that there are changes in the brain and in brain activity that will be being compensated in this period before symptoms. And if I could know what they were, then we could start to apply those more generally. This is just some data. The details aren't so important. It's just to say this is, again, this functional MRI, and we're just asking what are the fluctuations going on when the person's at rest. And the patients are in red. These are people who have ALS, they have symptoms, and the green are people who are totally healthy with no family history of ALS. And the blue ones in the middle are people who are gene carriers. They have no symptoms, they feel healthy, but they're a gene carrier. And you can see that they're not normal. They're not segregating with the healthy people, but they're not quite with the affected. And it's sort of an early clue, really, that there are subtle changes in the brain physiology that may well be helpful markers, particularly as we start to introduce some of these amazing genetic therapies for the rarer cases, and with much more wider applicability to non-genetic cases. So the last technique to tell you about is just this magnetoencephalography. It's a very sensitive way to look at brain activity. What you're seeing on the left here is someone making a decision to move. This is over 1500 milliseconds, and the brain activity goes up here and down in the visual area because you don't need to look once you've decided to move, although that's probably how accidents happen. But this person's making a decision to move, and that can all be recorded. And when we do that in ALS patients, we see when we ask them to move their right or left hand independently, we see this bilateral, both-sided activation, which is very unusual and suggests damage across the bridge, the corpus callosum. But when we do it in symptom-free genetic carriers, we also see early changes like that as well. And this is an article from Nature, just came out this year, which is showing that this kind of technology is heading towards being wearable. Now, of course, no one's going to expect someone to wander around with this on, but, this is just the first step. And I'm convinced that this is the sort of wearable tech that we'll be talking about certainly in a few years' time. And this is going to be real-time brain activity, which I think is going to help us unlock treatment in neurodegeneration. So David wanted me just to mention this sort of latest strategy, which is the multi-center biomarker resource strategy. I'm very proud of the acronym AMBROSIA, and it all came out of the Ice Bucket Challenge. You can see me just down here and my colleague Kevin Talbot. And it's a joint project with Sheffield and London. And what we're doing really is, what I want is every patient in the clinic to have the opportunity to contribute to research, because that's what my patients tell me they want to do. Now, not every patient wants to give spinal fluid, and not every patient wants to have an MRI scan, but most people want to do something, and a blood test is just as helpful. And I have to say the vast majority of my patients will do everything. But we want everyone to feel they can contribute, and so we collect all sorts of blood samples, urine samples. We have to label them all. We have to store them in liquid nitrogen. It's a big, big process, and we've had to integrate that into the clinic. The big picture is that we'll have all this data, genotype, clinical features, various scans, things we can measure. I haven't told you about eye tracking, but that's another marker. And we start to develop much more sophisticated models that we can use in a clinical trial to get a much earlier readout on what's happening with a drug, rather than simply saying, you know, are you less disabled after 12 months, which is a totally unacceptable state of affairs. So thanks for listening. This is my final slide. I am in no doubt that everything that I do relies on the great dedication of my patients. It's been an absolute privilege to see the efforts that our patients go to contribute to research. And what I feel that the very minimal thing I have to give them back is excellence in care. So that's always our priority. But it's a real partnership with our patients, and I have a great team in the clinic on the left here, but also a great bunch of people who do all the work. So thanks very much for listening.
Thank you so much, Dr. Turner. That's really appreciate that. That's such a great overview on the need for biomarkers. So a couple of questions. First of all, I think one of the questions that we often get is how people are working together globally to try to answer ALS and just to solve the disease, to understand it better. And I know that you've done a lot of imaging work over the years, and also they may know of here in Canada that we have a consortium called CALSNIC, led by Dr. Kalra. And I was wondering if maybe you might mention how the various imagers kind of integrate and share their information with each other globally.
Yeah, absolutely. No, the Neuroimaging Society in ALS is an annual meeting, and we started that 9 years ago. And actually Sanjay, who I know well, Sanjay Kaura from Alberta, was one of the early members and still comes along. And he set up the CALSNIC initiative in Canada. So we have a repository to share MRI data. That's all anonymized. So it's you know, these aren't scans that one could trace back to an individual, and we have consent to put those in a repository and share among us. And what that's enabled us to start doing is, you know, if I want to really understand how the disease is spreading, I need to take, you know, perhaps 50 patients who all have left upper limb onset. Now, I don't have 50 patients in my own clinic with exactly that type of onset, and I'd like to match them by age. I'd like to match them by speed of progression. And then we can compare and start to say, well, do we see the maximum changes in this part of the brain? Where do they move to next? And that's only something that we can do through collaborative work. So we've already started doing some massive studies with 200-300 scans, many of which came from Canada. And that's been a really, really successful initiative. I think, you know, the biofluids, you know, we're very used to sharing those as well. I have a collaboration with Miami, who do Michael Benatar, who does a lot of pre-symptomatic work. And again, none of us can get enough samples to do that on our own. But one of the great things about the ALS community is that we do generally share things very easily, and it works extremely well.
Great, thank you. Well, there's a question from one of the listeners about whether or not there's been done any tracking of white matter damage as biomarkers in pre-symptomatic mutation carriers.
Yeah, great question. Yes, there have. We did a little bit in some SOD1 carriers, and I wasn't confident enough of the results really because it was quite a small group. But subsequently, the Netherlands group, I mean, Jan was on earlier, actually, the same centre. They did a very large study in C9 carriers. And actually what they saw mainly was were grey matter changes occurring, but with some associated white matter changes. So there are definitely things happening before the onset, and I think that applies to all patients. So I think, you know, the landscape in the future is going to be about detecting system change in the absence of symptoms, and then giving medicines and having enough confidence to say, yeah, look, I know you might not feel, you know, that the medicine's necessarily doing anything now, but we can show from your scan that it's improving this. And so that's the way we're going.
Great. And one last question. First of all, I have to say it's super cool, the idea that maybe you could shorten the time between a general physician and getting to a neurologist who might be able to, then use tools to diagnose earlier by having a more generalized neurological test with neurofilament. I thought that was amazing. I just wanted to say that. But also, in terms of thinking about earlier diagnosis, there's a question about whether or not there may be existing databases in the world where people have provided biosamples and eventually they've come down with ALS, and those samples could possibly be accessed somewhere, or is that just too difficult to track?
No, that's a really good a really good comment, and exactly that is exactly what's been done. I mean, I used a hospital record linkage study to look at admission to hospital for other things before people got ALS, and that I think that was a way of tapping into associated diseases. But I think the study I would highlight the most is the AMORIS, M-A-O-R-I-S, Swedish study that came out in Annals of Neurology this year. One of the most exciting papers I've read. And here they had GP primary care blood tests for 30 years, going back to youth. And they had it in sort of millions of people and enough ALS patient converters in the few hundreds to then make a meaningful comparison. And when they did that, they found that the lipid levels, the cholesterol levels, and actually to some extent the glucose, the changing glucose tolerance over time, was really demarcated in the ALS people. So obviously no one knew at that stage they were going to go on to get ALS. But when you looked back, they had a different sort of lipid mix. Now, the really interesting thing is that that lipid mix is the so-called, healthy one, really. We have where we have more HDL than LDL cholesterol. So, whatever the result is, it's telling us something extremely important about the metabolic profile of people who are perhaps more susceptible to ALS. We often see thinner people. We often see people who are very physically active. We often get this hypermetabolism when the disease really kicks in, a lot of weight loss. We see weight loss sometimes 5 years before the first symptoms appear. So, those kind of databases are being mined, and there's more to come. There are secrets to unlock, and I think we're going to know much more very soon for people who aren't genetic cases about what the risk is.
Great. Well, thank you very much, Dr. Turner. We really appreciate you joining us today. That was wonderful, and we look forward to seeing how everything rolls out in the next few years.
Thanks very much for the invite. All the best.